Welcome to Why Individual Studies Can Be Misleading. In this section, we consider several reasons that the results of individual studies can be misleading or even wrong. We also consider how often misleading or wrong results make their way into the published research literature. Type 1 and Type 2 errors. Recall that researchers generally use null hypothesis testing to decide whether the relationship in their sample reflects a relationship in the population. The result is statistically significant, or whether it occurred by chance. The result is not statistically significant. Recall also that this decision is a kind of educated guess based on the strength of the sample relationship and the size of the sample. This means that no matter what decision the researchers make, it could be wrong. A type 1 error is when researchers decide the result is statistically significant when there is actually no relationship in the population. This is what appears to have happened in the flag priming study that you may have read about. A type 2 error occurs when researchers decide a result is not statistically significant when there actually is a relationship in the population. The point here is that any individual research result can be wrong even when the researchers have done everything right. Publication bias and the file drawer problem. When type 1 errors occur, their negative effect can be amplified by publication bias. This refers to a bias among researchers and journal editors in favor of publishing statistically significant results and again publishing non-significant results. Imagine, for example, an experiment on whether eating chocolate causes people to feel more romantic. Participants are randomly assigned to eat or not eat chocolate before completing a romantic feeling questionnaire. Imagine further that there turns out to be a statistically significant difference in romantic feelings, with participants in the chocolate condition scoring higher than participants in the control condition. The researchers are likely to consider the study a success and to submit it to a professional journal where the journal editor and reviewers are likely to find the results interesting and worthy of publication. But imagine that instead there turns out to be no difference. In this case, the researchers are likely to consider the study a failure and decide not to submit it to a professional journal. Or if they do submit it, the journal editor and reviewers are likely to find the results uninteresting and not worthy of publication. Publication bias is a problematic form from a purely logical standpoint. If a research question is worth asking, then the answer should be worth knowing, regardless of what it turns out to be. But publication bias also leads to the file drawer problem. While statistically significant results are published, statistically non-significant results are stashed away in research file drawers, nowadays in folders on their hard drives, where no one but the original researchers know about them. Thus, when you read a published study with a statistically significant result, you have no idea how many similar studies have been conducted with non-significant results. Researchers' degrees of freedom. It had also become clear in recent years that the problems of type 1 errors and publication bias are compounded by several fairly common research practices. Researcher Joseph Simmons and his colleagues have discussed what they call researcher degree of freedom. These are certain decisions that the researchers make during the research process, including when to stop collecting data, whether to exclude any participants from the analysis and which ones, which analyses to do, and which dependent variables and conditions to report. The idea is that researchers are generally free to try out many different possibilities in their research for statistically significant results. Another one is deciding when to stop collecting data. Some researchers look at their data periodically while they are testing participants. If the result is not statistically significant, they test a few more, look again, and repeat. If at some point they look at their data and the result is statistically significant, they immediately stop testing participants. Yet another researcher degree of freedom is including multiple dependent variables in a study, but dropping some of them when reporting the results. Imagine, for example, that we gave participants a romantic feelings questionnaire and also asked them to report the number of dates they would like to go on the next month, and to rate how much they enjoy love songs. If the results turned out to be statistically significant only for the number of dates, we might be tempted to write a research report that features that dependent variable, but does not mention the other two. A similar researcher degree of freedom is when researchers include multiple conditions in a study, but drop some of them when reporting the results. The problem with researcher degree of freedom is that if you analyze any set of data in enough different ways, you are very likely to find some result that is statistically significant just by chance, a type 1 error. In fact, Simmons and his colleagues showed that researchers who take advantage of all these researcher degrees of freedom in a relatively simple study can increase their chance of finding a statistically significant result to more than 60%. This is when there are no statistical relationships among the variable studies in the population. It's important to emphasize that taking advantage of researcher degrees of freedom to find statistically significant results, which has also come to be called p-hacking, does not necessarily imply bad intent on the part of researchers. On the contrary, this approach has been fairly common and accepted for decades. It's only recently that researchers have come to understand how large an influence it can have on type 1 error rates. The replication crisis. The problems discussed in this section have prompted many researchers to ask fundamental questions. To what extent do the results reported in the research literature reflect real psychological phenomena as opposed to type 1 errors? Certainly, to the extent that they reflect real psychological phenomena, other researchers should be able to replicate the studies and obtain the same results. But can they? Some observers have even suggested that the field of psychology research is experiencing a full-blown replication crisis. Many researchers have begun to address the issue of replicability directly. A group called the Open Science Collaboration organized a large-scale reproducibility project. A team of 270 researchers conducted direct replications of a representative sample of 100 studies published in 3 professional journals during 2008. One finding was that only slightly more than a third of results were successfully replicated. Another large-scale attempt to assess the replicability of psychological research was the Many Labs Replication Project. This large group of researchers replicated 13 classic and contemporary psychological studies in an online format with 36 different samples for each study. In this case, 11 of the 13 results were successfully replicated. One surprising outcome of this replication project was how variable the results for each study were across the different samples, even when they had used identical procedures. It seems clear from these initial attempts to assess the replicability of psychological research that many published research does, in fact, replicate. But many, perhaps the majority, do not. Regardless of whether this constitutes a crisis, it is clearly a problem. One final note is that all of the problems we have been discussing here, type 1 errors, publication bias, the file drawer problems, research or degrees of freedom, and replication failures are not unique to psychology. They affect other fields as well, especially the biomedical sciences.